Hello everyone, back with another video with another interesting topic. So today my topic will be on enzymes. So let us learn about enzymes today. Let us understand what are these enzymes, and enzymes are having an active site. What is that, and things like that. Let us analyze in detail. So what are these enzymes? Enzymes are protein molecules that act as a biological catalysts. They catalyze the chemical reaction. So, the enzyme acts upon a substance on which the enzyme acts upon, that one is called as a substrate. So, what is enzyme? Enzyme is a molecule, is a protein molecule which acts upon a substrate. Substrate is a substance on which the enzyme acts upon. So, there are two terms we are learning now. One is an enzyme, another one is a substrate. Enzyme is a protein molecule which acts upon substrates in order to form products. So, there are three terms: enzymes, substrates and products. What are enzymes? Enzymes are protein molecules which act upon substrates, resulting in the formation of products. So, they are biochemical catalysts. They catalyze the chemical reaction. Enzymes are the biochemical catalysts which catalyze a chemical reaction, in turn converting the substrate molecule into products. So, this is the introduction of enzymes. So, the molecules upon which the enzyme acts upon is called as substrate, and the enzyme converts the substrates into different molecules forming products. So, these are the things everybody should learn from the beginning in order to understand enzymology chapter. So, let us now, this is the introduction of enzymes. Coming to the different types of enzymes. So, enzymes are many types. Enzymes which act upon proteins are called as certain enzymes. Enzymes which act on lipids are called as certain enzymes. Enzymes which act on carbohydrates are called as certain enzymes. So, there are different kinds of enzymes depending on the substrate molecule they act upon. So, they are classified into three types. Like, different types of enzymes can break down different nutrients. So, what are these amylases? Amylases, they act upon starch molecule. So, converting starch into sugar molecules. So, amylase is an enzyme which acts upon a substrate known as starch molecule, in turn forming sugar molecules as the product. So, this is one example I am giving. So, what are these proteases? Proteases enzymes, they break down proteins. So, what are the products? When proteases enzymes act upon proteins, the products formed are amino acids. So, the end product of proteins, what are the proteins are made up of? Several amino acids. So, what will these proteases do? Proteases act upon proteins and convert them into amino acids, forming products. So, proteases are the enzymes which act upon substrates known as proteins, in turn forming amino acids. So, that is product. So, lipases. What are lipases enzymes? They act upon lipases, as the name itself indicates. The lipases, they act upon lipid molecules. That is, what are lipids? Lipids are nothing but fats and oils, converting them into fatty acids. So, what are lipases? Lipases acts upon lipid molecules, forming fatty acids and glycerol as their products. So, like this, there are different kinds of enzymes. There are like proteases, lipases, like that. There are different kinds depending on the type of substrate molecules they act upon. The enzymes are of several kinds. Coming to let us now understand how they are named. The naming of the enzymes. So, how the enzymes are named? Enzymes are named by adding the suffix known as ASE, ACE. proteases, lipases, like that. So, ASE to form the name of the substrate on which they act. Like in general, protease, ASE, they act upon proteins. Lipases, lipases, they act upon lipids. In particular, if you want to say like urease, which act upon urea. Tyrosinase, which act upon the amino acid tyrosine. Like this, the enzymes are named with the suffix known as ASE depending on the substrate molecule on which they act. Like this, the naming of the enzyme is like this. This is one way. Another way of classifying enzymes is that based on the type of the reaction they catalyze. So, for example, dehydrogenases. What does that mean? Dehydrogenases. The name itself tells the removal of water molecule. So, this is the type of reaction the enzyme catalyzes. Dehydrogenases means the removal of water molecules. Likewise, decarboxylase means the removal of carbon dioxide molecule. So, some have arbitrary names like pepsin, trypsin, etc. that comes in digestion process. Okay, now let us understand the active site of the enzyme. So, we all know that enzymes are the protein molecules on which they act upon substrate, in turn forming the products. These enzymes, they have a specific site known as active site. Only in that active site, that is the site where the substrate molecule can bind to the enzyme tightly. So, active site of an enzyme is the specific part of the enzyme. Only in that particular area the substrate can bind to the enzyme molecule in order to form the products. So, let us learn about the active site of an enzyme. This is very important. So, the active site of an enzyme represents as a small region at which the substrate binds and participates in the catalysis. So, likewise, the active site is the site. It has a specific amino acid sequence in the active site. Those amino acids in turn binds the substrate molecule tightly in order to form the products. So, let us understand what are the salient features of this active site of the enzyme and things like that. So, the enzymes we say, these enzymes are having secondary structures, tertiary structures like that. So, the existence of active site is due to the tertiary structure of the protein, that is enzyme. So, active sites are made up of, as I said, they are active sites, means there are specific sites which are made up of certain amino acids which are far from each other in a linear sequence. So, these amino acids in turn they help in binding the substrate molecule tightly so that it should form an enzyme substrate complex should be formed. After the formation of enzyme substrate complex, in turn later there is a formation of product molecules. Like that is, the active site plays a very major role and helps in binding the substrate molecule tightly to the enzyme, forming an enzyme substrate complex. Okay. So, the active site of an enzyme molecule, there are two parts. What are these two parts? One is the binding site. A binding site means it holds the substrate in one place. The enzyme has to hold the substrate molecule in one place. So, that is the binding site of an enzyme. Another one is the catalytic site. It is where the reaction occurs. So, the active site in turn it has two parts. One part is the binding site, other part is called as the catalytic site. So, these are the things that everybody should understand. So, So now let us understand with the diagram. What is this? This is the first one. This is the substrate molecule. This is the enzyme. So this enzyme binds. This is the active site of the enzyme. So the substrate molecule binds the active site of an enzyme, in turn forming the products. So these are the basics that students should understand in order to understand enzymology. So I think this video is helpful for all of you. Please study well. Take care. Bye-bye.